Hey everyone, welcome to my molecular biochemistry series. In this episode, we are focusing on transcription or eukaryotic gene expression. That is, we are going to focus on what is actually meant by a gene from a scientific model point of view, and we are going to think about some of the emergent properties that result from genes that give us as human beings, our unique traits and characteristics. And many people talk about their genes are the reason they are able to stay awake a long time, or play sports and be athletic, or in the medical field be the reason they have a disease or disorder. But before I jump into that, I do want to make a quick side note that I have officially begun production for my History of Mathematics Part 2. So for those of you that have been clamoring for that, it is currently in production, and hopefully I will get that out fairly soon. But back to the focus of this video, which is genetic expression. And so the first area of the gene and the genetic structure I want to look at is the promoter region. And as I work through this, keep in mind that a gene is just a segment or portion of a DNA strand, and thus it's just a bunch of different nucleotides in a specific sequence. Now, certain sequences represent certain components of the gene, but it's all just nucleotides and enzymes that catalyze reactions. So the promoter region is commonly in human cells displayed as cat teta, and those are just the nucleotides. And these nucleotides signal RNA polymerase II, also transcription factors, to bind to this region. And this will signal the beginning of transcription, which I have highlighted here with this green line. Now, following or downstream this transcription start point and the promoter region, we have all of our coding regions. Now, you have exons that will actually code for proteins, so those nucleotides will be part of the mRNA strand, the mature mRNA strand that will reach the ribosome to be translated. Other portions of this gene are called introns. These get spliced out in the second portion of transcription, which I will highlight in a bit. Then you have a polyadenylation signal sequence. This is AATAA, and this is where the poly-A tail will be placed, and this just signals pretty much the end of the gene. In addition, you have a silencer region that is made up of telomeres, which again is just more base pair sequences or DNA sequence that acts and functions as like a cap for this gene and protects the genetic information stored there. That is, protects the molecular structure and the nucleotides from being damaged or mutated, like we saw in my previous episodes. And it does this by capping the end of the gene. And so every time there is a replication in our cell before undergoing cell division, that telomere shortens and becomes less robust. And so the gene over time is going to be less protected. And that is why telomeres have been an important area for study in terms of longevity and aging. But do not worry, we have trillions of cells, thus we have plenty of backups if your telomeres begin to dwindle. And the last portion of the structure of a gene I want to emphasize and point out is upstream from the promoter region, and this is where regulatory proteins bind. This is how you get epigenetic modification, that is, without changing the code or structure of a specific gene, that is, change the nucleotides. You can add in, or the cell can add in, certain proteins that will either cause the gene to be expressed or not, and can silence it. And so this is the enhancer or silencer region, and these are important modifications, specifically epigenetic modifications to a gene. Now we can look at a simple template that really describes this transcription process the best, and that is once the RNA polymerase binds to the promoter region, the RNA polymerase will move from left to right in this case, and the template is this bottom strand. The mRNA is going to add its first base pair at the 5' end of its strand, which is going to be the 3' end of the template strand. And this pre-mRNA strand is going to be identical to the coding strand, except anywhere there is a T in the coding strand, the pre-mRNA strand is going to have a U. That is the only difference. And so we have our pre-mRNA. It's going to undergo splicing, sorry. And this is done with ribonuclear proteins. They splice out the introns. And you can have alternative splicing, and that is different arrangements of the exons. And this can actually influence what proteins are going to be translated. That is, what amino acids are going to be linked together in what order. So alternative splicing is relevant in that sense. In addition, during splicing, or right after splicing, another cap is placed. This is a methylguanosine cap, and this is going to be placed on the official or mature mRNA, messenger RNA. And we have our poly A tail, that's 200 roughly adenosines attached to the end of the mRNA strand. And then this guy will relocate to a ribosome, which are all over our cells, especially in the rough endoplasmic reticulum, and we are officially outside the nucleus with this mRNA, because all of transcription takes place within the nucleus of eukaryotic cells. And then the protein synthesis will commence. But I will end this video here, and I will see you guys in the next one. Till then, take care.